Start. Welcome to Third Eye Weekly. Joining us today is Dr.Eran from the Department of Genetics at the University of Leicester. Thank you for joining us.
Hello.
Hello.
Good to be at your show.
So Dr. Tauber is here to talk with us about the latest research unearthing 700-million-year-old DNA sequences from ancient animals and how they influence modern species. So tell us a little bit about what inspired your research into these DNA sequences of ancient animals.
Well, to be honest, we are not interested in ancient animals. We're interested in current animals, specifically current insects, which we're using in our research. And the idea is that in order to explore for important functional DNA element, you have to go and do comparison study. So the idea is that those sequences that are most conserved must be important, must serve some sort of a function, which is important for our research. So again, the idea is that we're actually interested in current modern animals, not really in the ancestors' animals.
So how does finding some of these ancient DNA sequences speak to how it's a little bit about the biology of our modern species?
Right. So the idea is that to look on conserved sequences. Now, when we talk generally about coding DNA, that's DNA that code for proteins. So most of our genes encode protein. That's actually easy task because we know the genetic code, and so even if you bring me some sort of a new DNA, I can, using computational method, I can easily see if any protein are encoded by this DNA. Now it's much more difficult. Most of our DNA is actually non-coding DNA. In fact, most of our DNA, it's actually under debate whether it functions or not. I mean, you would offer here that some people refer to this non-coding DNA as junk DNA because, again, it's not clear how much of it is important or not. So the task to find DNA stretches that are important in non-coding DNA is much more challenging, much more difficult. And so again, the concept that we're using is to look on many sequences for many species, compare them, and look on small stretches that are conserved. Now, the idea is not novel. We didn't like introduce any revolutionary concept. The idea of looking on conserved DNA is there for like a few decades. We just improved the algorithms and improved the statistics. So, and we got a small selection of sequences that we were ourselves really surprised how back in time they go. So we first were only focusing on a few insect species, wasps, honeybees. These are the animals that actually we work in the lab. And when we identify these conserved sequences, we try to look on further and further distant species, and it was quite amazing that a few of the sequences that we identified, came in such a huge evolutionary interval, So to date, two of the sequences that we identified go back like 700 million years ago, which is it is a record.
Wow, that's incredible. So what was the method? You used a method of phylogenetic footprinting, is that correct? How were you able to use this method in your research?
Okay, I'll try to simplify simplify the method, but in principle, first the first challenge is to identify similar genes in different species, which is actually not that straightforward. So you have gene X, and a snake might have also gene X that does this, that encode for the same protein, for example. It's a challenge to identify this gene X in you and in the snake. So the first task is to identify these homologous or matching genes. And then, of course, again, we're not interested in the sequence of the genes themselves. We're not interested in the coding DNA. We're interested in the non-coding DNA. So once we had these matching homolog genes, we extract the upstream non-coding DNA. And then just by matching this string, we're looking for strings of sequences of DNA that give us the best match. And this is done in a pairwise manner. So we started, for example, with a number of insect species. We compare 2 species each time, and again, we look for the sequences that give us the best match.
All right, interesting. So what were the animals that had some of the oldest DNA sequences, and what was the example, and what did that example show to the modern species?
Okay, so one conclusion from our study was that the most conserved sequences that we found they were placed next to genes that involved in a function which is translation of DNA to protein. So this tells us that this is one of the most important conserved functions that didn't change through all these years. Again, the function of translation of DNA sequences to protein. What is really interesting here is that we have these short sequences that are apparently important for control or regulation of protein that are important for translation, or again translation of DNA to protein. So again, this highlights one of the most important conserved function: translation of DNA to protein.
So you guys had a large interest in insects, as you mentioned. What were some of the insects that you looked at, and how was the conserved DNA in comparison to other animals?
Right. So our pet animal is a wasp. A wasp, which is called the jewel wasp, Nasonia. It's very tiny, harmless wasp. It's a parasitic wasp. And the reason that we're interested in that wasp is because the main focus of my lab is understanding the genetics of seasonal timing. So how animals know what season coming, and we know that the main cue for that, both in animals and in plants, is the day length. So animals know to distinguish between long day and short day, and they know if if it's winter or summer coming. So the main cue is a visual cue. But the problem is that we don't know anything at the molecular level. What is the molecular basis of this, day length measurement? And because this specific wasp had a very strong seasonal response, it's a fantastic model that we use to try to understand this process. But of course, we first need to understand the genome of that wasp so we can use it. So that was the motivation for the research. We wanted to really understand the genome of that wasp and to identify important sequences that will be helpful for us. So from there, actually everything started, because we started to compare the wasp to its relative. And the relative of the wasp, this is a group known as the Hymenoptera. So this group include ants, include the honeybees. So this is how things started. We started by comparing the genome sequence of Nasonia, the jewel wasp, to honeybees and ants, and then we identify these conserved sequences, and then we just added more and more genomes of different species, and just amazed to find that the conserved sequences that we found are present in such a broad range of animals. And of course, all the sequences that we identified are now available for the community in a public database. So now if a researcher is interested in a specific gene, all you need to do is just browse our database for your gene of interest and see if any of the element that we identified is attached to that gene, because this will give you like the first stepping stone to understand the regulation of your gene of interest. So hopefully the work will be useful not only for our research but for other researchers in the community.
So going back to the basis of your research, what are some of the sort of drastic responses that wasps have to the different changes of the seasons? And what's your hypothesis for something deeper than the obvious external changes that come with the seasonal changes?
Oh, really? I'm so happy that you asked this question. So again, in general, it's kind of a black box. Understanding how animals, including human, tell the difference between the seasons. The idea with the wasp, so what happened with the wasp is that the female detect the shortening of the day in the autumn and the coming of the winter. So like many other insects in temperate region, the female will generate progeny. This is a parasitic wasp, so the female will lay eggs in the host, which is like a pupa of a big fly. The progeny usually will develop, but because if it's a short day or winter is coming, the progeny will go into developmental arrest, and they will not develop. They will just wait, sit there in the host, waiting for the right moment to develop. And they can wait for like 1 year or 2 years. So they're in a state that we call the diapause. And again, this is actually present in many insects in temperate region, like in the USA or in Europe. What is really fascinating about the wasp is that it's actually the mother that detects the cue, detect the day length. And somehow, in a way that we don't understand, the mother transfers the information to the progeny in some molecular way. So because the response is made by the progeny, they decide whether to develop or not. So this is one mystery that we really want to understand. And research in my lab, you can do a lot of things with insects that you cannot do with human. So for example, we can, if we have some candidate genes that we suspect that are important for this process, we can silence. We can knock down these genes and see what happened to the response. There's a lot of area that is studied in my lab, which relate to epigenetics. So there's a lot of, I don't have time to go into it now, but there's a lot of process where the DNA is being tagged by other molecules that detect whether decide whether these genes will be expressed or not. And the wasp is a fantastic model to understand this process of epigenetics. So this is another area that we study. And of course, the main thing is that this daily measurement represents one type of clock that animals have, which is the seasonal clock. But on top of that, we have another clock, which is also really the focus of my lab, which is the body clock, what we call the circadian clock. This is a clock that drives daily rhythms, 24-hour rhythms, because as you may know, your body is changing throughout the day, all the time. So, if you measure blood pressure, temperature, level of hormones, all these things are changing throughout the day, and this waves, oscillation are driven by the body clock. So basically, you have two clocks. You have the body clock, the daily clock, and you have the seasonal clock. Now, one of the $1 million question is whether these two clocks are linked together. This is like under like a huge debate. And so we try to carry experiments to see if indeed these clocks are connected together. And of course, we focus on at the molecular level, whether you need the same genes for the body clock. Do you need the same genes for for seasonal timing, which again is an open question. But yeah, we hope that the WASP will be a great model for that. It's quite a new thing for us, because another insect that we use in the lab, which is our main model, is the fruit fly, Drosophila. So most of actually what we know about the body clock came from studies in Drosophila. So and it's amazing, because the same genes that are important for the clock, the same genes that are present in Drosophila, are present in mammals, including human. So Drosophila was really very useful for understanding the body clock. The only problem was that, for us, was that Drosophila doesn't have a very strong seasonal response. So that's why we introduced the WASP into our lab. And we hope that we would be able to develop and apply the same technique that we have for Drosophila. We hope that we would be able to develop the same advanced genetic methods to use with the WASP as well.
All right, awesome. So what do you perceive as some of the future research that you guys want to go forth with?
So again, we obviously most interested in the two most conserved sequences that we identified, those 700 million years old sequences. What is amazing is that these two short stretches of sequences are associated with two proteins, yes, but the two proteins actually work together. So these are two proteins called ribosomal proteins. So we have ribosomal protein 1 and ribosomal protein 2. And each of them has this very ancient, important conserved stretch of DNA. And we know that these two proteins interact together, again, in regulation of translation. So we're really keen to, start doing some experimental work to dissect exactly the role of these stretches. And of course, with fruit flies, it's easy. We can do, the sky is the limit. We can delete these sequences, or we can modify the sequences. We can generate transgenic flies, and we can see the impact on translation. We have the knowledge, we have the instrumentation, we have the techniques, we have everything. We just need to get someone to do the work.
All right, awesome. Well, that's all the time we have today. Thank you so much for taking the time to